Can you click the OK? Wait, nothing's come up. It's all hidden.
Senior faculty there.
It sounds really familiar. It's on here. It's right. You look up there. There it is. Yeah. Thank you.
Yeah. And he's written.
Okay.
Yes.
Okay, so I think we're going to get started. It is my great pleasure to introduce our speaker today, Dr. Hazel Barton, who is the Loper Endowed Professor in Geological Sciences from the University of Alabama. Dr. Barton's experience is on cave microbiology. Very, very cool. And I'm just reading her little bio on her page, and it's just spectacular. I'm very, very much in awe. Her research is geared towards understanding microbial interactions and processes in cave environments. She's got over 100 papers published. I've got a whole bunch of public outreach and media. You've got BBC, NPR, Animal Planet, National Geographic, part of an IMAX movie. Wow, Journey into Amazing Caves. That's so cool. She is a fellow of the NSS. She is a Kavli Fellow of the US National Academy of Sciences. She's a past chair of the Committee on the Status of Women in Microbiology for the American Society of Microbiology, which is very cool. And then lots of awards, lots of funding, superstar that I am just very grateful that she has come here to talk to us today about cave microbiology. And I'm going to open up the floor to you to take it from here. Thank you so much for coming.
Thanks, everyone. I know that you guys have a lot of speakers. You have a very active program, both in oceanography and astrobiology and the other programs you're doing. So I know it's a lot of places that your time gets taken up. So I appreciate everybody coming to see my talk on cave microbes. So I'm going to just kind of jump straight into it. Oh, I hope so. There we go. And talk about why caves. So I did have a whole, usually when I talk to a lot of graduate students, I have a whole history of my background and how I ended up getting here. And then I was putting all these slides in, and I was like, as I added more science slides, I took out more and more of the history slides. So I'm more than happy to answer questions on that. But the brief synopsis is when I was 11 years old in comprehensive school, it's called in the UK, I got to look through a microscope at pond scum, and that was it. I was a microbiologist. That was like, I drew everything I saw. And it was only 4x, so there wasn't a lot to see, but it was just amazing. And then when I was 14, I did an Outward Bound course, and they took a bunch of city kids. My parents were completely non-outdoorsy. First time I ever slept in a tent, I think I was 19. But they took us out into the woods and let us go feral, and they rounded us up. But we did rock climbing and horse riding, kayaking. And they took us caving, and I was just, I don't know what it is about this place, but it just feels special to me. So when I was 16, there was in England, you do our A levels. If you ever seen Harry Potter, there's advanced wizard level. And we had electives on Wednesdays to do whatever we want. And a teacher took us caving. And I pretty much until I became a professor went caving every weekend. And then when I became a professor, I had to write grants and papers. That kind of wasn't maybe once a month, once every other month, but I had a really amazing advisor, a guy called Norm Pace, who's a pretty famous microbiologist. And he said, there's a time to be a caver and there's a time to be a scientist. And right now is your time to be a scientist. And that's very true. Sometimes I'm very much into caving, doing lots of expeditions, getting fit, getting strong. And other times I'm sitting behind a desk, fat, drinking too much coffee, blah, blah, blah. So, but when I was in graduate school, I went caving every weekend, and my advisor said that caving it's going to ruin your career because you're spending too much time on it. And at that time, it was in the 90s. I got my PhD in 97. You had to keep your science and your personal life kind of separate because it was felt that one diluted the other. But now that siloed approach has very much gone away. And the reason we have such interesting, innovative science is because people can bring their own ideas and background and experience to it. And I think that's the true meaning of diversity in science, like why it's important. It's where we have different ways of looking at things. So I look at caves as a microbiologist. Caves are interested in a biological context because they're deep in the terrestrial subsurface. It's basically a portal into that biosphere. Obviously, there's oxygen everywhere. That's the one thing. It's oxygen is coming in. That's why we can go there. So you are looking at an oxidized surface in there. But the deepest cave in the world is 2,400 meters deep. So you can go a long way into terrestrial subsurface. And a lot of times you're looking at, as you get deeper, they're getting more isolated, and you're looking at how energetics change. That geologic isolation, a lot of times when the processes that form a cave stop, that cave becomes static. And you can see microbiological effects that have taken thousands, tens of thousands, even millions of years to develop. And that way it's a great analog for astrobiology because you've got surfaces that don't weather. There's no wind, there's no rain, no sunlight that breaks them down. So you can actually understand what some of those biominerals look like. And I have a big NASA Mars sample return idea that I'm really interested in doing with an atomic probe, but maybe not. We'll keep pushing that one. They're nutrient limited, usually levels of organic carbon, incredibly low, ultra oligotrophic. And the nitrogen can be in the sub ppb level. So nitrogen fixation and acquiring nitrogen is really important. And they're low biomass, so less than 10 to the 5 cells per gram. Because of that, I actually get to be on a lot of the Mars 2020 and a lot of sample return committees, because they're like, how do you do things without contaminating? How do you know you're looking at the microbes from this environment and not something you've been introduced? To talk about the caves that we do, why Alabama? Alabama has 9,000 caves. So when they offered me a job there, I was like, usually they struggle to find, they're like, oh, will you please come? And I was like, it's probably a terrible negotiating tactic, but I was like, yes, want to go. So 9,000 caves, they're really interesting geology. Most of the caves have a 200-foot entrance pit to get into like the cave proper. There are pits in Alabama that are over 400 feet deep. The caves that we study, we generally study some of the deepest and longest caves in the world. This is a picture from Cheve. This is on the way to Camp 4B, I think, in Cheve. It's 3 days to get to that point where that person is carrying that pack, and everything has to be hand carried when you're in the cave. Lots of rope work, lots of technical rope work. The rope work I actually find a lot easier because I cannot fall to my death when I'm on a rope. I mean, you can if you do it wrong, but if you do it right, you cannot. Like, it's that when I'm free climbing, I'm like, I'm gonna fall to my death. So I do actually really enjoy the rope work. I used to enjoy the splashy, but then I've been flooded into a cave 3 times. And if you guys ever have seen the Tide Cave rescue stuff, it's a little bit traumatic. And because we're cavers, we are actually cave explorers as well. It's kind of like, I'm very fortunate in that people come from all over the world to do research in my lab because they have a dream of being a professional cave scientist. So I get just amazing students, but we're all very technically proficient. And because of that, we can actually either be the explorers. My postdoc and one of my graduate students are pretty high class climbers and A climbers, or we can go directly above the climbers. This is my friend Derek at the top of a 435 foot pit that they climbed. And we can go in and do the microbiology. And that allows us to sample things when we know there's been no anthropogenic impact on those sites. So we did a study that showed that antibiotic resistance was ancient because it's almost impossible to find anywhere in the world where you can say that humans have never been and antibiotics have never gotten, but thousands of feet down in a cave, it's entirely possible to do that. So we have a lot of projects in our lab. I'm going to talk about the first 2, deep aquifer microbiology and manganese oxidation, this near infrared and low light phototrophy, because there is photosynthesis in caves. Who is a fantastic agent in geologic change. I kind of did have that in there, but I was really going to have to squeeze it in. So I decided not to rush that. But you asked me about poo. I am an expert in poo. iron reduction and iron ore by mining. So this is a project we did in Brazil showing that caves can actually be formed by microbial iron reduction. And we actually, the microbes that live on stalactites and stalagmites have to deal with very toxic levels of calcium to make a living. And so the way they do that is they actually fix atmospheric CO2 to make a carbonate to detoxify the calcium as calcium carbonate. And we figured out that there was actually a really important carbon sequestration process there. We do have an NSF grant that looks like it's going to be funded as of today, but I don't know, maybe tomorrow. But so my students have rolled out a startup company and are leveraging that. And anybody that's not aware of the I-Core program who has an idea for something that they're doing could be valuable is an industrial approach. You know, do look into the I-Core, especially if you're doing anything with metals right now. So I'm going to talk about the deep aquifer microbiology. So we do this work at Wind Cave in South Dakota. It is somewhere that I went as a graduate student. It's one of the places that my advisor told me I was wasting far too much time in. It's in the Black Hills. It is a national park. It is known for its buffalo, but underground, Wind Cave National Park, it's actually this cave here. So this is, I think we're the 7th longest cave in the world right now. It's 163 miles. I started coming here as a graduate student and just exploring. So go up, pretty arduous trip through it. Most of the passages you walk for 20 feet, then you crawl for 20 feet, then you climb for 20 feet, and then you repeat that. This is an area that we recently discovered, but it's a pretty mazy cave. In the 1960s, some cavers discovered this passage here, which is formed by a big joint, a big fault in the rock. And they followed it down to this area here and found a series of lakes. And these lakes are very different than lakes you would find anywhere else in that they are formed where the cave intersects the aquifer. So normally, if you think about a cave is made by rainwater. Rainwater flows in the entrance and it works its way down to the water table, and then there's more cave down there. This cave is pretty ancient. It's one of the oldest caves in the world. The cave forming processes that form the cave we're into today, it's probably over 9 million years old. So it actually is older than the Madison aquifer itself. So the Madison is a huge aquifer that underlays most of Wyoming, a lot of South Dakota, up a little bit of Nebraska and up into North Dakota, and 2 million people get their water from it. It's very difficult to study the microbiology of the aquifer because it's quite deep. It's about 100 to 200 meters deep. And the way that you sample it is usually through wells that are drilled down into the water. The trouble is that wells, when you drill them, they go through all the rock between the lines, the aquifer and where you are. There's often aquifers, higher level aquifers within that rock. And also they case the wells. So if they want the well to go down through the rock and not collapse, they'll case it with something like steel. So when you're sampling the microbiology of the aquifer through the well, you're actually sampling the microbiology of the aquifer and the microbiology of the well casing and the microbiology of the rock and the microbiology of any aquifers that are higher up in the rock series. There's a sandstone rock over the top of the limestone that the cave's formed in. And when we sampled the aquifer through this well, what we found is pretty much the microbiology of the Middle-Eocene formation. So a lot of people have been writing papers about the microbiology of the Madison, but they weren't really studying the microbiology of the Madison. The cave, because it's older, the rock here, the passages are older than the Madison. What happened is when the water table rose to its present level, it basically flooded the cave. So when you're going in there, you're not going into kind of a bathtub that's filled with water that's rained down from the surface. You're going in and you're looking down on the potentiometric surface of the aquifer. Getting there, quite challenging. So this is my undergraduate student, Riley. So she's in this passage here we call the boxwood chimney. And actually it's about 40 feet high. These fins here we call boxwork. They're very rare, the forming caves. They're formed by a condensation corrosion that happens to be very common in Wind Cave. And these things can be 3 feet long. And if you slip, they will cut you like a knife. And then the passage is quite small. So everything we do, we have to hand carry through these passages. So we can't, there's no backing the truck up and then pulling all the gear out, right? Everything has to be small and everything has to go in the cave. We have this thing that I came up with when I was a brand new faculty. It's called the Magic Box, and we still have the same container. It says Magic Box on the top. And the Magic Box is a little box, Tupperware, and everybody, when they go on a field trip, has to look through the Magic Box and they have to add something to the Magic Box that's not already in there. So I've seen safety pins and needles and twisty ties from bread bins, bread bags, you name it. Somebody has to put something in there that isn't already in there. Because when you're 3 days into a cave and you get there and you realize you don't have a thing, you cannot go to the car to get the thing. So you get out the Magic Box, and when you open the Magic Box, the thing is right there, just like magic. So Magic Box. The lakes, this is looking down on the surface of the lakes. This is one of my undergraduates. We're 127 meters underground. It takes, for an experienced caver to get there, about 2 hours. Most of my students take about 4 hours to get there. It's pretty treacherous. There's some really interesting climbs. The oldest water using isotopic analysis in the lake is 50,000 years old. The youngest water is 30 years old. So it takes a long time to get from the surface to these lakes. And the organic carbon is really low, 0.2 milligrams, 0.29 milligrams per liter. And I think that carbon is all autochthonous. So I think it's all from autotrophic growth. The research I'm going to talk about is my graduate student. She's now Dr. Olivia Hershey. She works at MIT. And she did all of the metagenomic. A lot of the stuff she did was figuring out how we sample the lake. So when we went down there, we're going to do microbiology. And a lot of you guys are oceanography people, so you're familiar with doing microbiology, or at least somebody who does microbiology in ocean. So you get a liter of water and you filter it and you do the microbiology. We went down, we got a liter of water, we filtered it, we did the microbiology, and we got no DNA. It's like, we messed that up. So we went down and we took a filter with a pump and we filtered 10 liters of water. And then we came back with the filter and we extracted it and we got no DNA. And then I was like, okay, we have to do this the proper way. So we went down, we collected 3 times 10 mils of water and counted the cells in it. When we did that, we found out that we have one of the lowest biomasses of any water that ever been analyzed to date. So the open ocean, you're 10 to the 4 to 10 to the 7. You know, a lot of these reservoirs, Crater Lake, which is considered oligotrophic, all the way down to Wicca, and we're about 2,300 cells per mil. So we were like, this can't be right. We must have messed this up. So we went down, we collected more samples, and we collected twice as many as we needed. And we sent some to my friend in Germany, who is an expert in low biomass cell counting, and he got the same number. So one of the lowest, well, you've got Lake Vostok, which is not, this is the accretion ice, so it's not technically lake water. And then you have this stuff that Thibaut's lab did in South Africa. So that's kind of up to 3,600 meters below the surface. And it's that really hot, poor water, cracked water that comes out. So as far as the lake, we are one of the oldest. So to figure out what we needed to how much we needed, we back calculated, right? So if your average cell is like 1.3 femtograms of DNA, if we want to have 500 nanograms of DNA to be able to do a metagenomic library, we have to filter at least 400 liters of water. It turned out to be able to get this to work, we actually had to filter 1,200 liters of water to get enough DNA to do the work. What made it really challenging is when we looked in there, it turned out that there's a very high percentage of something called the Petesky bacteria. If you're not familiar with them, there's 2 schools of thought on what the Petesky bacteria is. Originally, it was thought that they were a unique radiation within the bacteria. So somewhere between a deeply branching, somewhere between where is the archaea on here? It must be down here.
The down right down the bottom.
Oh yeah, there's the archaea. Yeah, between bacteria and archaea. But none of them have been cultured. And they're all ultra small, 0.1 micrometers in diameter, which probably explained why we're having a hard time finding them because we're using a standard 0.2 micron filter, which is what you use for water microbiology. And it turns out 10% we're shooting through. There's a new tree that came out. I think this is still an argument that they're just a unique radiation within the bacteria, but still, as far as I'm aware, no cultured representative and tiny. We took a sample. We had collaborators at the Joint Genome Institute. And we took a sample and they did some single cell whole genome sequencing on the ultra small cells. So you can pick out the ultra small Patesky, and you can pick them out because when you use a DNA primer for the RNA genes, they don't stick very well in the Patesky because they're so divergent. So anything that doesn't glow using regular 16S primers, you can actually isolate with single cell sorting. Then you put that in a separate well, and then you sequence that bug on its own. And they did that, and it turned out we had incredibly small genomes. And these organisms, again, when they measured them, were very small. They were all below 0.2 microns in diameter. And they also didn't have the regular central metabolism like a PCA cycle, tricarboxylic acid cycle. So it turns out that these guys just kind of do fermentation, right? They're very small and they don't need a lot of energy, and they seem to do most of that through fermentative pathways. They don't even use respiration. When we went to do our 1,200 liters of sampling, this is our pump. These are a bunch of batteries that we had stacked. These are backup batteries for laptops. And we were able to get 24 hours of pumping. So we'd have to go in, do all of this, set it up, come back out, wait, and then go back in to get the 1,200 liters. This is a filter that's used in dialysis, and it actually has a 30 nanometer pore size, so we can catch those really small cells. One of the things that we did, I thought that was very clever of Olivia to come up with, is like, well, what does the big population, right, the regular size cells, and this small Patesky bacteria population look like? So what she did was she filtered it, plugged the filter, and this is her extracting it. You just this tube in, back extract it, and then she actually put it through a series of filters in the cave. So when she came out, she had a filtrate that was above 0.2 microns, between 0.2 microns and 0.1 micron, and then below 0.1 micron. If you look at the whole community, it was incredibly diverse. So this is where there's no separation. It's the raw cells, very, very diverse. The big, fat, juicy cells, we saw an enrichment in species like the proteobacteria, which are the big, fat guys that everybody's aware of. But when we looked at the small population between 0.2 micron, we saw an enrichment in certain clades. And these were the Patesky bacteria, which are these ultra small cells that we imagined would be there, but also the nitrospira. To explain what's going on here, this black line is what the relative proportion is in when you don't fractionate them based on size. The brown is when you look at the fat cells, and the gray is when you look at the tiny cells. So we saw an enrichment in the Patesky bacteria, which is what we anticipated, but also this massive enrichment in the nitrospira. And using genetic probes, it turns out these nitrospira are about 20% of the entire population down there. We saw enrichment in Verruca microbia, which are also ultra small cells, and then nanoarchaeota, which are also ultra small. So it looked like 40% of the cells in this community, not only is it incredibly diverse, but they're also very, very small. And this is something that we've never seen before. And the other thing is we thought we would see just a few specialists, right? There's no organic carbon. There's hardly any energy there. Like, why is it so diverse? So she looked at the nitrospira to try and understand what the nitrospira, why were there 20% in there? And what she did was she assembled some metagenome assembled genomes. So I think she was able to generate like 11 from this population, but she was able to find 2 within the nitrospira. This nitrite oxidizing organism, and then this class trogloglia, which was actually just it was described. If we'd have been a year earlier with this paper, we would have gotten a Nature paper. But this is the paper that Jared Ledbetter's group discovered that are manganese oxidizing autotrophs. So very closely related to this group of organisms. It was postulated that manganese oxidation was capable of supporting a microbial community, but never demonstrated. And Jared has this really cool story where the undergraduate was supposed to wash the dishes and left the dishes on the side of the dish cupboard for like 3 weeks and forgot about it. And when they went back, one of the graduate students noted that the manganese carbonate had turned black. And they were able to isolate this bug out of there. And that's where they found manganese oxidation. You just had to wait a really long length of time to see this stuff happening. And then you get Nature paper. We looked at the energetics that support this diverse community within the cave. We did see autotrophy, but we didn't see rubisco. We saw reverse TCA cycle. So just in this incredibly starved environment, right? You can only do the rubisco pathway. You have a lot of energy to power it. We saw a lot of metabolism of C1 and C2 compounds, which I thought was interesting. But the thing that just kind of, smacked us in the head was the amount of manganese oxidation. So our nitrospira is probably carrying out autotrophic manganese oxidation. So it appears that this community is driven by manganese autotrophy, so fixing CO2. To determine if that was the case, I had a master's student, Jane Waddle, and she did a lot of comparative organic chemistry within the lakes versus the rest of the cave. And this is excitation emission matrix spectroscopy, which helps you actually identify the kinds of organic carbon that you found there. And it's almost exclusively bacterial based. So we have an ecosystem that is driven by autotrophy, and the autotrophy seems to be manganese oxidation, which is the only place this has ever been described where we have a community that's dominated by manganese oxidation. And that's pretty hard to do because almost everywhere else you have iron. And when you have iron oxidation and manganese oxidation, you have these absorption, and it's a very, very complicated and difficult to detangle those two pathways, where we've got an entire ecosystem that's driven by that. I asked one of my colleagues who's a geologist, I said, do you have any idea why there would be manganese oxidizers dominating this lakes and wind caves? He was a geologist. He spent his whole career studying these caves. He's like, oh yeah, there's tons of manganese there. The subaqueous helictites are manganese. And I was like, that's what these things are. They're subaqueous helictites. They're pretty big. The biggest one, if I was standing next to it, it's probably about this high. I saw them when I was a graduate student back in 1993. And I said, oh, that's a microbial biofilm. And everybody was like, no, you say everything is microbial because you're a microbiologist. And I'm like, that looks like a fossilized microbial biofilm. And I'm sorry that it's not as bright as you can see on my computer, but it's super intricate and beautiful. And they had this idea that it was common ion effect and like carbonate-rich water was coming up and there was kind of mixing and then it was precipitating. And I was like, and then Art says the core of those things is manganese oxide. And so when you slice them in half, you can see these cores of manganese oxide and they're full of microfossils. So my graduate student who's now my postdoc, George Bruley, he's attempting to use a lot of techniques to demonstrate that these are actually the microbial fossils of manganese oxidizers. All of a sudden, we're getting excited and we were proposed to the P-STAR program because they have very similar manganese oxides that they found in Gale Crater. So this now gave us a link for a biosignature. Can we understand these? And these very intricate structures as well, we thought, well, maybe if we send a robot to Europa and it's able to go through the ice and start swimming around, it's got a camera, maybe it snaps some pictures of some weird things that looks like that. We've got like this nice big biosignature. Okay, so these subaqueous helictites are found. Remember I talked about the cave being on this big passage, being on the big joint that goes down to the lakes there. They were all found along that joint. There's Riley, my undergraduate, sitting next to some of them. You see now it's a bit more obvious how they're coming out of the floor. And what I thought is, if there's a big fault in the limestone, so this is the Madison that the cave is formed in, it has an aquifer. There's a big crack in that limestone that goes all the way to the lower levels of rock. Underlying it is something called the Deadwood Formation. And this is a silicate that's full of manganese. And so it's 2 to 3% manganese. And I thought maybe there was a crack through the limestone to the deadwood. The aquifer, the deadwood, could bring that manganese up through this crack into the cave, which is why you would see the formation of these bushes all along that fault. So this is the model we have. You have a lake, and it's on a crack, and that crack allows places for the water to kind of ooze up from below. That water is rich in manganese, and you have manganese oxidizing biofilms form along that crack. So in that way, it would be like kind of like a hydrothermal vent that you see in the ocean. Instead of being driven by sulfur, it would be driven by manganese. Okay, yeah.
Manganese inclusions, do you see any kind of fluorescence or reaction to UV light?
I haven't looked. Is there a reason?
In Mammoth, we were finding a lot of stuff with different mineral inclusions creating different fluorescence effects, especially in the fossil deposits.
Yeah, we haven't. We can take a look at that. Yeah, we're kind of going to do some weird stuff with the manganese oxides. We want to try and get a biological signal to see if the manganese oxides are biogenic, not just here, but throughout the whole cave. But we can look at that. Think about these lakes. Okay, so now we have a baseline for the lakes, manganese oxidizers, supporting microbial community, incredibly nutrient limited, incredibly diverse, incredibly low biomass. When you look at the structured microbial community, it's 97% bacteria, 3% archaea, and no eukarya. There's no eukaryotes, right? If you find a scant spore from a fungus, if you use a fungal stain, but they're so, like, you have to filter 10 liters water to even stand a chance of seeing one. So we think they're getting blown in. But we found a ton of bacteriophage. So what you can do is you can actually filter out the bacteria and then continue with this filter that's 0.01 micrometers and look and see there's like a starry night, and those are the bacteriophage. And this is one of the SEM images of some bacteriophage. So there's hardly any biomass, and the literature suggests that when there's hardly any biomass like that, you shouldn't have any bacteriophage because there's not enough to support it. And we're finding 10 to 1, which is why you find in the oceans. It's what the standard is. So why are there what is driving so many bacteriophage in there? Because the bacteriophage are essentially, this is me being funny, but the top predator in these lakes is a bacteriophage. So my graduate student, Nate Friedrich, is now doing his PhD on the phage within the lakes to try and understand why so many phage within there with the low biomass and low nutrients, right? Phage kill the winter, right? Is the diversity we're seeing because there's so many phage killing the winter? And if they are, why did the phage play a role in keeping the community healthy? He started with a metagenome. So the first thing he did is take all the metagenomic data we had and look for the presence of phage. And created this chart from, I think he did like 60 different environments. Are they more temperate, which means the virus goes inside the cell and carries genes with it. And if the cell gives it a good home, the virus gets replicated as the cell gets replicated. So there's a selection pressure for the phage to keep the cell happy. And there's a reason why the cell would keep that phage. So those would be genes that would be actually supporting the cell keeping the phage. There's kind of a mutualistic interaction between the phage and the cells. To me, I like the idea of the phage being kind of like cloud computing, where you're living in this very challenging environment. And if the environment changes, you have this big database of additional accessory genes that you can kind of pull in when there's an emergency and adapt. The other is lytic, where you're more lytic because there's lots of energy and you just want to replicate as fast as you can. You don't kill the host because then you just absorb onto another host and make more of yourself. So there's kind of bursts of phage. And we sit almost exactly on the line, which didn't tell us anything about what was going on. But then he did an analysis. So this is all of the different phage populations he did using this metagenomic approach. And the blue one are temperate phage. So these are the ones that go inside their host and kind of go along for the ride and keep the host happy and they get replicated. The green is lytic, which is where we want to burst it and it's spreading like flu virus, right? Infect everybody as quick as you can. And then the red is we have absolutely no idea what they are. So we are right here. So not particularly useful that we're seeing that. That didn't tell us anything. So then he looked at the accessory genes carried by the phage. And so this is looking at what are the phage carrying inside the cell, and maybe that tells us something about benefits that they could be carrying with the cell. And there were some interesting things like secretion, motility, chemotaxis, and then intracellular trafficking, things that we think could benefit a cell if environmental conditions change. But you see how they're hatched. That's because less than 1% of the population of phage we identified carried this information. So there's just not, it's not telling us anything. It's just noise. I can get up here and present it and sound like this means something, but it is basically meaningless. And the reason is that when we did the metagenomic approach, we're taking all of the bacteria and we're breaking them up and looking at the DNA that's present. The only phage we're going to catch are lytic phage that are absorbed to the surface and carried along for the ride. They're going to be lytic cell phage inside the cell replicating before we bust the cell open to get the DNA out, or they're going to be temperate phage inside. What we have to do is we have to study that free floating population, those 10-to-1 cells that we see. We have to purify the phage and then look at what is actually in that kind of cloud of information to see what they could be contributing to the bacteria, because you're not going to capture that with this snapshot. So these things are tiny. Collecting, the average bacteria is 4 million bases of DNA, and that's 1.3 femtograms of DNA. And the phage, the average phage carries about 40,000 bases. So these things are tiny and they carry very, very little information. So it's very hard to get the DNA. We ended up doing phage plates, and I used to work in a salmonella lab and postdoc. And you never, you will carry stuff from your postdoc and you're like, I'll never need this. But I had a whole phage system from when I was a postdoc. So Nate actually grew up phage, titrated them, and ended up with a solution of phage that had, I think he had 10 to the 10 phage per mil. So what we can do is with those phage, he can start doing his filtration extraction techniques, and we can figure out, can we actually get enough water to be able to do these extractions, and how much is that? This is how much, right? So if you can see, this is a 100 microliter pipette tip. This is how many phage, using this approach, we would get if we filtered 2,000 liters of lake water. So this is really hard to do. So I've got another oceanography joke here. So you guys can put one of these in the ocean, right? This is what we have to carry everything through to get to the lake. So we're 4 hours from the entrance, and we've got to fit everything through there, which you don't have to take your helmet off. You can't see, the reason he's trying to stay high is because there's a big pool of water there, and if you go upside down, you'll get pushed underwater and you won't be able to come back up. So everything's got to go through that. But luckily, there are some aerospace people. So I don't know if anyone's familiar with Stone Aerospace. They've done a lot of work for Europa. And the CEO of Stone Aerospace, Bill Stone, is actually a caver. And he and I were on an expedition together. And I told him about this problem he had. And he said, why don't we write a grant together? So we wrote a grant, the P-STAR program, for BEAGLE, which is the Biomass Enumerator and Geochemical Limnological Explorer. It's very good. It's actually named after the boat, that Darwin's boat, Beagle. And this is our artist conception of what it looks like. It's going to have a big antenna that sends signals through the rock. It's going to hunt around and look for things to see if it can find the microbes we're looking for. And then if it finds them, it's going to send a signal to a cell phone tower that we're in, or a repeater that we're putting above the cave. And that's going to text me and say, come get me. It has cell counting that can go down to 50 cells per mil, and a manganese electrode that actually allows us to do a lot of electrochemistry in the cave. But manganese is an absolute nightmare because it absorbs the surfaces. This was the first prototype, so we wanted to see if the filtration would actually work. So this is the first. It looks like a big dinghy. I'm sorry, it's black on black there. Maybe should have picked a different color. But this is George with it in the kiddie pool on its first expedition. Can tell you the kiddie pool has more bacteria in it than the lakes at Wind Cave. And then this is the updated version because it turned out that the filter that the entire world has been using to do phage in the ocean has holes in it that allow most of the phage through. And so we did the study that showed 97% of the phage actually get through this filter and aren't captured, which is fine when you're in the ocean because you have so many phage, but we can't lose a single phage. So we had to go back to that 30 nanometer cutoff to do that. Now, the actual chassis is made from pieces that fall apart. Remember, they've all got to go through that hole we have to go through. So the widest bit can only be 8 inches. So it takes apart so we can hand it through. And then each of these are going to be big floats that it floats on. Each of these floats is actually engineered with the components that allow us to have a sonde that goes up and down and send all the signals to the surface. And then I have a student, Anna, and she is using our MAG data, the metagenome assembled genomes, to actually develop unique culture media to culture some of the ammonia, manganese oxidizers. These are just an example of some of the manganese oxides that we find within these caves, just meters thick and super slimy. We actually think they may play a role in formation of the caves. So in the last few minutes, I want to shift to a story I'm only going to spend a few minutes on because we're getting towards the end of the day here. And that is the near-infrared low light phototrophy. This is a work of my graduate student, Assyria Fiji. And so this is just based on, I have to explain to you guys about main sequence stars, and young stars have different kind of light, and old stars are cooler. So their light is redshifted and their luminescence goes down. So our sun is pretty rare in the galaxy. Only 7% of the stars are young teenage stars like ours. Most are in K and M type stars. So when we look at the potential targets to look for the presence of life, there's a lot that don't fit the conditions that support photosynthesis on our planet. So is there somewhere we can go and look for the potential for photosynthesis in a environment that might mimic a K or an M type star? And of course and then something that looks like these stars, and we can say these can support photosynthesis or not. Of course, it's a cave talk. You can find those conditions in a cave. The low light is pretty obvious. My friend Lars approached me when I was giving a lecture in Copenhagen and said, can we go look for photosynthesis in caves? So I was like, oh, bless your dear heart. And then I gave him a whole thing about how caves are dark and there's no photosynthesis. And he's like, well, yeah, let's go look. Okay. So we went to Carlsbad Caverns. This wall is completely black. There's no sunlight that hits this wall. This is the main entrance. The sunlight comes down and hits the far wall. And we went and we flashed our lights up in here, and the whole thing was bright green. What was happening is the sunlight was hitting the far wall and then bouncing back. And so we were seeing, and it was super bright green, like a super vivid green. And then we went further in the cave. So now you can see, you can kind of see we have our headlights on, because you can't, you can kind of see, but it's that twilight, at night when you have to get your phone out to see where you're going. Still photosynthesis. And then this is in the dark zone where you can't see your hand in front of your face without your light on. And there's still green stuff going there. When we looked at the light, we saw that the amount of light at these different sites, obviously as we were at the entrance, it's pretty bright, 1,000, about what you see with direct sunlight, significantly lower in this twilight zone. And they were still photons, but they were few and far between in the deep zone. But the really interesting thing is when we looked at what that light looked, rather than the photosynthetic active spectrum, which is 400 to 700 nanometers, when we looked at the near infrared, we actually saw an enrichment in the deep zone. So for some reason, even though there was a lot lower flux, we got a lot more of these near infrared photons. And I was like, you know, I love rocks, what's going on? I remember that light bouncing. And I was like, well, what wavelengths of light get bounced by limestone rock? And it turns out once you hit 700 nanometers, those photons get bounced more. They get absorbed by the rock up to 700 nanometers. And above that, they get reflected. So a cave is essentially a hall of mirrors to near infrared light. So it's bright, visible light at the entrance, and as you get in deeper, it gets enriched for this near infrared. We were seeing 600 times more near infrared light in the dark zone of the cave than we were seeing in the entrance. So incredibly enriched. And these are starting to get the luminosity and the wavelengths of these K and M type stars. This is a cave in Borneo. We're about half a kilometer from the entrance here. I just told the photographers to turn their flashes towards the far wall and take a photo. And they did, and you can see how green everything is. And as soon as we did this, everybody was like, oh my God, it's green in here, right? Lots of photosynthesis. So I just put an ICAR proposal together with a whole bunch of folks, which may have been a big exercise in futility, but we did it anyway. We didn't know at the time. But basically what we can do is we can 3D map these caves and actually measure the wavelength and flux of photons at each site, and then build a ray model based on that and figure out what position, where the sun is in different positions in the sky, what would the average amount of light be hitting any particular area in that cave over the course of the year. And then we can see something that models our sun with light hitting Earth, our sun with light hitting Mars. I think this is the TRAPPIST system, the LHS system, and then maybe even the limit, like how far can you go? And we can go a really long way. You can go in, around a corner and half mile down the passage and still see evidence of photosynthetic activity. Isuru started trying to culture some of the bugs out of these caves. So just racks and racks and racks of tubes. These things take a very long time to grow. And he actually, I was like, get some really cool bacteria. And he got a really cool algae. So this is our algae. We isolated this from a site in a cave where we've measured the wavelength, the dominant wavelength at 730 nanometers and the flux at 0.03 photons per meter squared. So when you think about how much light hits the entrance, which is 1,000, you can see we're, you know, many orders of magnitude below, right? So this is a very dark environment. So that's able to support photosynthesis. This is oxygenic photosynthesis. This is what we're really focused on. Because if you want to look for the potential for life around another planet, you want to be able to try and detect oxygen. I know that there are many ways that oxygen could be around those planets that's not biogenic, but it's a fantastic signal. And if we can constrain what is possible based on Earth, then we have a potential for like maybe point this telescope here. There's lots of weird things. So in the twilight zone, you see these pinnacles form. It's me looking at them. And they all point directly towards the light, the entrance. These things can be 1 meter long. And there's a remodeling of the rock that we're trying to look at. It looks like it's the cyanobacteria kind of boring into the rock. And the reason this is interesting is I did an expedition to Greenland, and we were looking for carbonates way up here. So we're 82 degrees north. This is my friend Gina. She does paleoclimate work, and she had a grant to go look at the caves in this massive here. You can see one cave entrance there, one cave entrance here. We were camped down here, and it's really quite challenging to get up there. We did have a helicopter for some of it. This is looking out of one of those entrances. So it's just like being on Mars, incredibly cold, incredibly dry. And the caves are very, very cold themselves. The average temperature of the caves are -9 degrees centigrade. We were in them down to -19 degrees centigrade, and we were still seeing photosynthesis, which is below the theoretical limit for photosynthetic activity. And we saw these same things. We saw those same pinnacles. We have a nice biosignature that could be preserved in carbonates for this. And I know I'm at time now, so I'm just gonna, looking for life on planets, the potential for maybe figuring out if there are alternates for oxygenic photosynthesis. You can have, maybe there's a red edge and you can actually see the reflection of a different kind of photosynthetic pigment around M and K and M type stars. You're going to be tidally locked. So the front might be super hot, the back might be super cold, but maybe there's an equatorial region where the conditions are just right to support photosynthesis. And this is my favorite, is maybe the bit pointing towards the star is way, way too aggressive and nasty, but maybe there's an aurora on the backside of the planet that has enough light to support photosynthetic activity. And then if you have James Webb, what we'd like to do is say, point here, because we know those conditions could support it. So when I started, caves were only known to be formed by carbon and sulfur, and my labs added iron, manganese, we think nitrogen, and I was not going to put phosphate on there, and I was talking to Kevin today, and I'm like, I'm going to put phosphate back on my slide. So we've tripled the number of ways that caves can form, and thank the folks in my lab, our sponsors. This is the Stone Aerospace folks helping us out at Wing Cave. The staff at Wing Cave who are super supportive of us being there and asking them to build buffalo fences so the buffalo don't knock down our antenna. And then all the cavers that lug the heavy gear through the caves for us. So thank you. Yes.
I have two questions that are completely unrelated. One of them is, have you sliced open the pinnacles and seen if there's any interesting structure in the middle? And the other is, how are you going to avoid contamination with your floating probe?
Yeah, both excellent questions. And I was rushing through it because I was at time and I was like, I misjudged it. Those pinnacles, if you imagine this is the surface of the rock, those have been cut back. So it's actually the original surface is the tip, and then they've been cut back by erosion. When we slice them and look down, you just see regular limestone rock, but what you see is all of the cyanobacteria just having a go at that rock. There's definitely a lot of red algae on there. We can see a lot of red algae going on. And to me, that makes no sense because there shouldn't be enough, like reason we focused on cyanobacteria because there shouldn't be enough energy there to support something as big as the eukaryote. So why are the algae there? And that's the question that I think, like I think we really are interested in these novel pigments and how far can we take the near infrared. That's really like our whole group is mostly chemists to try and figure out like what is the limit for light limit for light, but then at those low wavelengths, what you can support. So yeah, the algae thing to me is like, it's so tempting to pursue it, but I really think that the question I have is going to be more easily answered in cyanobacteria because they can tolerate so much lower levels of energy. And then how do we, well, that is actually really important for P-STAR, right? Because the idea is that these instruments will eventually go to, Europa and Enceladus, wherever and land. So we actually have to build this whole thing so it can go in the autoclave. So we're like a Viking type approach. Yeah. So it is a trick. We've got to do all of this. I've told them I don't want any lubricants or organics or anything exposed on the surface that can drip into those lakes because they're so nutrient limited, right? We don't want to change that chemistry, and we can't introduce any bugs because those are the bugs we have found in caves that the cave environment is so specialized that we've gone and looked because some people have argued, well, you shouldn't even be in here in the first place because your microbiome is contaminated. And we've gone to some of the sites where people camped and actually gone through the sediments and looked. And after a few months, there's no evidence of humans. And I think what it is they just can't make it. And everybody else just comes in and goes, whoop. It's all the, it is, we carry all the waste out, right? You don't leave anything in. When we camp on these big tarps. And so it's a big piece of plastic. So any hair or skin cells or anything is on there, food crumbs. And we kind of put it all up in a bowl and take it out with us. So we do a lot of stuff to limit that. And it is a concern. I've seen pictures of people wearing Tyvek suits to do this, but I tell you, absolutely, that is just for the photos. Because when we were doing the white nose syndrome, I was working with bats with white nose syndrome. One of the groups required us to wear Tyvek suits to go in and out, so we didn't contaminate anything. And that entire Tyvek suit was just a trail of pieces of Tyvek between the entrance and where we just took it off. I mean, it's, if you see someone in a cave with a Tyvek suit, it is purely for the photo, so. Yes.
So these manganese oxides, are they, so the manganese oxide is microbially induced or precipitated?
Yeah.
But then you also have carbonate around.
Oh, yeah. Sorry for that. So think, right, so you got the water coming in from below and you have these streamers that should be doing this in there and flow. Eventually that lake, the water's gonna stop coming in, right?
Yeah.
So the potentiometric surface kind of sinks away of the aquifer. So there are parts of the cave that are higher and drier, and we think about 200,000 to 1.3 million years ago. And those things are classically there, right? The ones in the lakes now we won't be able to see, we need the robot. And what happened is when the potentiometric surface left, there's no more recharge coming in and the lakes now become static and they slowly dry out over time. And as they dry out, the carbonate concentration gets increased, so they stick to every surface. So all of the basins, you can actually see when you're in an old basin because you have carbonate precipitate. And also those things happen. I suspect it was pulsing, like it came up and went down and came up because you actually see bands of it like a tree. So I think it took a while. And so you may have had a bit that's still flapping around at the top, but over time, eventually the whole thing got fossilized by the carbonate.
Oh.
Yeah, it's kind of cool. Some of it's really thin. And you saw one of the ones where it's very, very thin and it's still super black. Another is like, it's fatter than my finger. Yeah.
I was curious, earlier in your presentation, you mentioned antibiotic resistance.
Yeah.
The low energy cave environment and how isolated they are. Is that part of the reason why they're hard to culture just in the normal place? And then also just wondering if you had any like medical professionals contact you or try to collaborate with you on investigating the bacteria in cave and space out like that antibiotic resistance and their like unique nature.
Yeah, so my PhD is actually medical microbiology. So yeah, don't tell anyone in the geology department. So we found that they were resistant to every known class of natural antibiotic. So the only things they weren't resistant to were synthetic or semi-synthetic antibiotics, which makes sense, right? Because they wouldn't have been exposed to them. There was a really, really high amount of them. Like almost every organism had multiple, not just one or two resistances, like, 7+. One had 28 different kinds of antibiotic resistance, 9 of which had never been described before. And so we were very excited because it meant that you can actually know what that might look like in the clinic in 20 years when it emerges and already have ways of defeating that, right? Clavulanic acid is a great way if you've ever had augmentin, right? It's augmented penicillin. So yes, we have worked together and we've published, and I've worked with some really talented people on that. And that was kind of a highlight. Like, it's funny. I feel like one of those bands that has a hit in the 80s and then everybody asked them to play that hit, right? I know that sounds terrible, right? We got this paper out and I was really excited about it on the time, but then I moved on and now it's like manganese oxidizing and thing, phototrophy. This is all really excited. And I still get reporters that call me up and ask about this, but it sounded terrible to say that, but it actually ended up in the textbook. And that I think was a highlight of my career as a microbiologist, to do a thing and then be teaching undergraduates and then flip to a page in the textbook. And I know it was in there because they asked me for the photo, to see a photo of a cave and then an explanation that antibiotics are ancient because of this study. Yeah, it was kind of cool. Yeah, but I still feel like a bit like an 80s band. So I'll do those. Don't Stop Believing soon. Yes.
I hate to ask the same related question. Production in that environment.
We haven't looked at that. It's super hard to do antibiotic production because they get assembled very differently. Antibiotic resistance is easy because what you do is just expose them and see if they're resistant. And then you can actually go and look for those gene cassettes because they're quite defined. But for making novel antibiotics, a lot of these are their non-peptide ribosomal synthases. So they build them like peptide chains, but they don't go through the ribosome. And you can have 26, 27 different components. If you look at an antibiotic, they all look like a spider's web because you got a billion years of evolution of tinkering to make it work perfectly. So it's hard to spot them easily. And actually finding novel antibiotics is challenging for that reason. There's programs you can use like anti-smash. I can't remember what it's antibiotics is the anti, and then I can't remember what the acronym is for smash, but it will pull them out. And it'll tell you not so much if you have novel ones, but if the pathways are like known or if there's something that maybe is a little bit exotic in there. I have a colleague, Brian Backman, that does it, and he exclusively looks at antibiotics in caves. And we did find a novel one, and I was all excited to get that published. And one day I'll resurrect that paper and get it published. But it was a beautiful new molecule, but it had a penicillin backbone. So nobody cares, right? It's like something completely new, but it just looks like penicillin. You can't patent something that has a penicillin backbone, even if it's new. So there's probably thousands and thousands of novel ones they've come up with, but no one's going to spend any time or money investigating it unless it can be a drug. So it's a really good question. It's a terribly waffly answer, but I hope I answered it for you. Yeah. Yes.
Yes. Really fascinating talk. I have a questions about the decay photosynthesis.
Mm-hmm.
So is there some way to tell whether photosynthesis is the main energy source for these microbes, or are they also doing other things as well? Like is
Right, I know. Yeah, I hope the reviewers kind of like don't think to ask that question, but the fact you did means I'm probably screwed on that. So yes, the biggest thing I'm worried about is that if we look for the limit, like, are we seeing a limit of where we're just seeing the presence of the genes, or is it the physical limit of where oxygenic photosynthesis can happen? And so I think the key to that is to look at the other structure of the community using metagenomics and what kind of level of heterotrophy. And so I'd like to err on this is definitely an autotrophic system. So the dominant process here is autotrophy versus we're seeing such a mixed bag. Are these species, right? Because all they can be is like blown in and we're going to be detecting them. And it's not really a photosynthetic community. It's just contamination. The other thing is we're going to be using digital droplet PCR, which is super, super sensitive, right? So we only need a few cells to give us a false positive on that. So we did spend a lot of time thinking about what that structure could look like and where we would draw the line. And I think what we would do is we'd be super, super, super conservative until something convinced us otherwise. So it'd have to be more than just simply cultivation. Some of those sites where you see like green, that's only going to be photosynthesis, right? That's not contamination when it's that green. And we're at such low levels of light there. I think we are a long, long way from knowing where that limit is. So we might be talking about, the difference between 0.003 and some crazy number where we might detect species there. So it is a big, like with Asuru's algae as well, he keeps trying to present, it's like, we've got blah, blah, blah. We've not grown it in the lab at 0.03 micromoles of photons, right? We've grown it in the lab at 730 nanometers. We know it's redshifted, but if we try to grow it under that light flux, we're going to be feeding it now, and he's going to be feeding it when he retires, and he still may not have a colony. So there's a lot of questions there, and it is a complex question, and it's an important one. Yeah. I'm sure we'll get it wrong multiple times, and somebody will ask that question many, many times. Yeah.
How many of these have you been able to culture in a lab, and are you able to like run any lab experiments with these bacteria?
So these are algae?
Just like in general.
Oh,
like,
these? Yeah, we got thousands of them in the fridge. Yeah, yeah. Grow 'em. I hate Bucaldaria. You're the Bucaldaria guy, right? Yeah, yeah, yeah. We got lots of things. The algae we've got this. It looks like we got the same species in 3 different locations. So I think it's a real thing. Yeah. Yeah.
What do you use to culture the manganese oxidizing bacteria?
Culture, liquid cultures. Yeah, so we are basically just trying to do what Jared did, where they had the flask on the sink side of the sink. So we're trying to do that. The key is to have like a high surface to volume ratio, so plenty of oxygen gets in. We did try bubbling oxygen through it, but we didn't see anything that way. I think the key is light. I think that you see manganese oxide crusts in caves all over the world, but not on surfaces outside of caves. In fact, there are some cave entrances where you're following a stream passage out and it's completely black from all the manganese oxides. And as soon as you hit the zone where the light kind of cuts in at a certain time of day, it just turns to rock. So I think light might be really important. Yeah.
So I see that we've gone over the time, so I just want to thank Dr. Barton one last time for an amazing tour. Thank you. Do you have a few minutes to stick around? Because I know there's lots of really interesting questions, so I don't want to like shove everyone out the door. I want, please come talk and ask questions. But thank you again for such an amazing.
Thanks, everyone.